Okay, welcome to Chapter 11 in Gunstream, and today we'll be talking about blood. So we're going to discuss the general characteristics of blood and describe its function. We'll talk about erythrocytes, those are red blood cells, and describe the normal concentration of red cells in the blood. We'll explain what hemoglobin does. We'll explain where the production and destruction of red blood cells takes place and factors that control this. We'll describe the types of white blood cells and what each of them do. We'll indicate the normal concentration of white cells in blood. We'll talk about the structure and function of platelets. We'll talk about the structure and function of plasma. And we'll discuss the concept of hemostasis and explain the basis for human blood types and why this is relevant in a clinical aspect. And we'll identify the blood typing antigens and antibodies in each ABO blood type, and we'll describe some major blood disorders. So blood is a liquid connective tissue, one of two liquid connective tissues, the other being lymph. And we have what's called a closed circulatory system, which means that our blood flows through either vessels or through the organ known as the blood pump, the heart. Not all animals have a closed circulatory system. Insects, for instance, have a substance similar to blood that bathes every organ in the body, as opposed to our system in which the blood vessels come into close contact with almost every single tissue type. The functions of the blood, including carrying substances throughout the body, so it's a transport mechanism. Examples include oxygen and wastes. It regulates the pH of body tissues, and it prevents excessive blood loss as a result of hemorrhaging in a process known as hemostasis.
And we're going to take a look now at the
So blood is confined within the heart and the blood vessels. We have what's called a closed circulatory system, which means that our blood either flows through vessels or through the blood pump known as the heart. Not all organisms have a closed circulatory system. As an example, insects have a fluid very similar to blood that bathes every organ in the body. But we have blood vessels that come into close contact with almost every conceivable tissue. The functions of blood include carrying substances throughout the body, so it's a transport medium. Examples include oxygen and wastes and hormones. It also helps to regulate the pH of body tissues. Remember, that's critically important because if the pH of the body tissues gets too high or too low, will cause proteins to denature, and the activities that those proteins mediate will break down, among those being chemical reactions. Will also change membrane potential. And remember that membrane potential is a method that cells use to store energy in order to perform certain tasks. So blood, very important in maintaining the pH in most fluid-filled compartments between 7.35 and 7.45. It also prevents excessive blood loss due to hemorrhaging through a process known as hemostasis. Hemorrhaging is just a fancy word for bleeding. And it also helps to fight infections. We'll find out that it's the leukocytes, the white blood cells, which are able to generate a physical and chemical attack that keeps us pathogen-free and disease-free. Blood is a fluid connective tissue. Now, how do we know that it's a connective tissue? Well, if you look at blood under a microscope, what you find is relatively few cells and a lot of material between the cells. In this case, the material between the cells is a fluid known as plasma, and thus this qualifies blood as one of the two fluid connective tissues. It's heavier and more viscous than water because it has substances dissolved in the plasma, like proteins, waste products, ions, and so forth. Viscosity is simply a term meaning resistance to flow. For instance, maple syrup is extremely viscous, whereas water is not. Blood pH ranges between 7.35 and 7.45. Typical male blood volume is between 5 to 6 liters, while females have between 4 to 5 liters. It's approximately 8% of our total body weight. Now, you may be wondering, why is it that men have more blood than women on average? And there's a couple of reasons. Number one, men tend to be larger. In addition, men tend to have a greater muscle mass. Women, by the same token, tend to lose blood as a result of menstrual loss. It's also true that men have more testosterone than women do, and as a result, their hematocrits tend to be higher. So those are several of the reasons for the difference. 55% of blood volume is plasma. That's the liquid between the formed elements of blood, which we call the cells. 45% is the formed elements. Those are the erythrocytes, the leukocytes, and the platelets, known as thrombocytes. Most of these are red blood cells, which carry oxygen and to a lesser extent carbon dioxide around the body, while 1% are white blood cells and platelets. Now, white blood cells, we're going to find out a little bit later on, fight disease using a variety of physical and chemical attacks on pathogens before they get a chance to spread throughout the body. Platelets, as we're going to find out, assist in blood clotting, and this is so that when you have a puncture, a laceration, or an abrasion, you don't continue to bleed indefinitely. Red blood cells are also known as erythrocytes. The prefix eryth means red, and cyte means cell. Transports oxygen and, to a lesser extent, carbon dioxide around the body. Now, why is this important? Well, oxygen is critical because cells utilize oxygen to make a large amount of ATP in a process known as aerobic respiration. In the presence of oxygen, a cell can produce between 36 and 38 ATPs per every molecule of glucose that it takes in, but without oxygen it can only make 2. So this is why we need oxygen delivery to every cell, every tissue in the body. Carbon dioxide is one of the end products of aerobic respiration. It's a waste gas, and we have to eliminate it, and we do so through the urinary and the respiratory system. We can't let this accumulate too much in the tissue fluids and in the plasma because it would cause the pH of those fluids to drop, and this would produce a chemical situation around and near the cells that again could potentially denature proteins and will perturb membrane potential. The red blood cell is unique in the body. It's one of only a couple of cells, the other being the thrombocyte, that has no organelles and as a result lacks DNA. So there's no nucleus, there's no mitochondria, there's none of that. Most of the volume of the red blood cell is taken up with the oxygen-carrying pigment hemoglobin. The biconcave shape is critical because it promotes improved diffusion through the membrane. So we have a lot of surface area there to get oxygen in and out of the red blood cell. Remember that it is basically a transport vehicle for these gases and also a way to deliver gases to different body compartments. So mature red cells lack a nucleus. This is the most numerous formed element. If you look at a stained blood smear, you'll see these little round discs populating most of the slide. Those are the erythrocytes. There's hundreds and hundreds of them in a typical microscope field on the lowest power of magnification, which would be the scanning objective. Hemoglobin makes up a third of the red cell volume. It's a red pigment, and it contains heme, which is an iron-containing pigment. The heme is actually an organic molecule with an iron atom in the center. The molecule looks a bit like a spider web, and it's mostly carbon and hydrogen. And then the globin, which is the protein component, which is what holds the heme in what we know as the active site of hemoglobin, which is where the oxygen binding is going to take place. Now, the oxygen-carrying hemoglobin is termed oxyhemoglobin, while oxygen-deficient hemoglobin is termed deoxyhemoglobin. Oxyhemoglobin has a bright red color, and this is due in part to the fact that it's bound to oxygen. It's also due to the numerous double bonds in the heme and the iron atom itself. There's a property of different solutions. Solutions that contain metal ions tend to be brightly colored, as do solutions that contain numerous conjugated double bonds. They tend to be brightly colored as well, and that's true in the heme region of hemoglobin. Oxygen deficient hemoglobin tends to be less bright red. It tends to be more of a dusky red or a dark red, and this appears blue when we look through the systemic veins at the fluid that it carries. Okay, so your blood never really is blue. It just appears blue as it courses through the veins, which return the blood back to the heart. But that dark red color is indicative of an oxygen poor state for that pigment. Okay, red blood cells are the most abundant formed element in blood. Red blood cells are counted in a particular volume of blood to determine the oxygen carrying capacity. And so what we call this is a hematocrit. And the way we do this is we take whole blood and we put it in a capillary tube, a little small thin glass tube. We plug both ends with clay, and then we spin it in a centrifuge, and then the packed red cells represent that percentage of the volume that are the formed elements, the erythrocytes. The higher that level of packed cells is, the higher the hematocrit, and as a result, the higher the oxygen carrying capacity. We can also do a count, a clinical test to determine the red cells in a cubic millimeter of blood. Adult males have 4.5 to 6.2 million per cubic millimeter, while females have between 4.2 and 5.4. Again, the difference is due to the difference in size, the difference in testosterone levels, and the menstrual loss that we see in females. Okay, so that's why the difference in these averages exists. Males have a higher metabolic rate, and this requires more oxygen. That's due primarily to the greater muscle mass. Muscle is a metabolically active tissue. In order for it to do its job, it needs a lot of oxygen, and so we need more blood in order to deliver the oxygen to the muscle tissue as it performs its activity. Altitude influences red count. You tend to have greater red counts for individuals that spend a significant amount of time at high elevations, and the reason for this is the body is compensating for the lower percentage of oxygen in atmospheric air at those elevations by increasing the number of red cells so that it can more effectively bind the oxygen in the atmosphere at that elevation. And so we call this condition polycythemia. It's normal in individuals that live up in the mountains. It's abnormal in individuals that tend to spend most of their time at sea level. Okay, prior to birth, red cells are made by the liver and the spleen, but after birth, the red bone marrow takes over this task. In infants, red bone marrow is in all bones, but in adults, it gets restricted to the skull bones, the ribs, the sternum, the vertebrae, and the pelvis, primarily in the area populated by spongy bone. This is hematopoietic tissue. So here you can see where blood cells are produced initially and how we respond to changes in oxygen level by changing the amount of red blood cells that we produce. If blood oxygen levels are low, erythropoietin is released by the liver and the kidneys. This stimulates the red marrow to make more red cells. When oxygen levels reach normal, the erythropoietin secretion depletes, goes down. This decreases red cell production. These low levels of erythropoietin are usually present to maintain basal levels of red cell production, but again, they respond to changes in oxygen concentration so that we never have a situation in which the circulatory system can't deliver sufficient oxygen to tissues in order to support their activity. Other factors required for red blood cell production include iron, which is needed for hemoglobin production, and folic acid and B12, which are needed for DNA synthesis during red cell formation. B12 absorption from the diet requires a protein called intrinsic factor that's secreted by the stomach lining and allows the B12 to be absorbed across the gastric mucosa, which is the inner lining of the stomach, and directly into the bloodstream. All red cells develop from hemocytoblasts that are present in red bone marrow. This is a type of adult stem cell. How long do they live? Well, about 120 days. Okay, so you're talking about 4 months for the average lifespan of the red cell. About 2 billion are destroyed and made every second. The worn out red cells are removed by phagocytes, which are in the liver and the spleen. Heme is broken down after this process. It breaks into the iron-containing compound heme and bilirubin. The iron gets recycled to form more hemoglobin, while the bilirubin is secreted by the liver into the bile, which enters the small intestine, where it serves a detergent function. Basically, it causes emulsification of the fats in our diet that come from the stomach to the small intestine, and this allows the lipases that are produced by the pancreas to be more effective at their job. White cells, known as leukocytes, are formed by hemocytoblasts in bone marrow. These cells retain their nuclei, so they have DNA in them as well as cellular organelles. There's about 4,500 to 10,000 per cubic millimeter of blood, so there's far fewer than the erythrocytes. The white count increases whenever cells encounter disease-causing organisms or chemicals. We call these pathogens, as some disease-causing entity. A pathogen can be a living entity, such as a bacteria, a mold, or a fungus, or a parasite. It can be non-living, such as toxins and viruses. And this is just a diagram showing you where the different leukocytes come from. So the hemocytoblast stem cell is in the marrow, and it can go down one of two lineages. It can produce a myeloid or a lymphoid stem cell. In the case of the lymphoid stem cells, these cells migrate to special areas of the body. The lymphoid stem cells that are going to eventually produce the B lymphocytes, they remain in the bone marrow, while those that are going to eventually produce the T lymphocytes migrate to an organ known as the thymus. The lymphoid T and B cell precursors eventually give rise to precursors to the mature T and B lymphocytes. And once that's produced, what will happen is that these cells will stay in lymphatic organs, such as lymph nodes, the spleen, to some extent the liver, and in the connective tissue of the body. And then when chemically signaled by an injury or an infection, they will travel to that site and do battle. Okay? The myeloid line will eventually differentiate into either red cells or platelets, or a special type of white cell called a granulocyte. The granulocytes come in 3 flavors: the erythrocytes, which contain red staining granules, the basophils, which contain blue staining granules, and the neutrophils, which contain pink staining granules. Normally you can't see these granules. It's only when you treat them with a blood stain that you're able to pick these up. These have different roles in the body. The eosinophils tend to respond in allergic reactions and battle parasites. The basophils mediate the inflammatory response by releasing histamine, which is what's contained in these blue granules. And the neutrophils fight bacterial infection via chemical attacks and phagocytosis. Okay? The platelets, as we've already discussed, mediate blood clotting, while the erythrocytes are there to carry oxygen and carbon dioxide. Down here, the lymphocytes generate chemical attacks that help to destroy a particular type of invader. They can use a variety of methods. They can use chemical sprays that cause infected cells or pathogens to kill themselves, or they can use a device called an antibody, which is what's produced by the B lymphocyte, which is a protein that will bind to and inactivate, or in some cases signal the destruction of that particular pathogen. The macrophages are important because they wander around the body looking for infected tissue or pathogens. They eat them via endocytosis, basically phagocytosis, and then they have a lysosome fuse with that endosome, chop that particle into little tiny bits, and then present it on the surface of the macrophage cell membrane like a wanted poster to warn the lymphocytes that there is a particular pathogen inside the tissues of the body, and that we should engage in destruction of that pathogen by causing the lymphocytes that are best suited to destroy that pathogen to multiply and then go out and do battle. So let's talk a little bit about the role of the different formed elements, what their task is, and how they do it. The formed elements, particularly the leukocytes, provide disease defense against pathogens, and they regulate the inflammatory response. Now you might ask, okay, well, what's the inflammatory response? Think about the last time that you had a serious injury, a cut, a puncture, an abrasion, and what happened immediately after the injury. The area reddened, it warmed up, it swelled up, and it was probably somewhat painful. This is the inflammatory response, and it's designed to bring blood to the area of the injury so that the white cells can begin the process of fighting off pathogens, removing dead tissue, and then ultimately repairing the area around the injury. So the cells perform their function within the tissues. They can migrate from blood into tissue and from tissue into blood, and they do this as a result of chemical cues that they experience in the environment. They follow a chemical trail to the site of the damage, and then when they locate the area of the injury, different cells perform different functions. Leukocytes can be grouped into two broad categories, the granulocytes and the agranulocytes. The granulocytes are called granulocytes because when they're stained, they possess visible cytoplasmic granules, and they come in three classes: the neutrophils, the eosinophils, and the basophils. The agranulocytes have cytoplasmic granules that are not visible upon staining, and these include lymphocytes and monocytes. So when you look at them under the microscope, you won't see any granules present in the cytoplasm. That doesn't necessarily mean that they're absent. Now, let's talk a little bit about neutrophils. They make up 70% of the total white cell population in typical sample of plasma. The nucleus has 2 to 5 lobes and has lavender staining granules in the cytoplasm. These tend to be the first white cells that respond to tissue damage. They phagocytize pathogens. They destroy necrotic tissue. They fight bacteria, and they release an enzyme called lysozyme. Lysozyme is a chemical that weakens bacterial cell walls by essentially cleaving the sugar bonds that hold the bacterial cell wall together. The result is when those bacteria experience an osmotic shock, they'll either rupture or they'll shrivel away without the protection of the cell wall. So very important that you understand that. Their number increases when you have an acute infection. So in this sense, this is useful as a diagnostic tool because the doctor can take a blood sample and can compare your neutrophil count to a typical neutrophil count and determine, based on that information, essentially what it is you might have a problem with, what condition you might be dealing with, whether it be bacterial infection, if we have Other types of granulocytes that have elevated counts, the diagnosis will be different, and we'll work through those as we work through these different formed elements. Eosinophils, upon staining, have reddish granules. They make about 3% of the total white count. They have a bilobed nucleus. Their function is to neutralize histamine release during allergic reactions, so they are essentially very important in turning down the immune response. They destroy parasitic worms, so they're parasite fighters, and they tend to increase in number also during allergic reactions. So what they do there is they help to complex and destroy antigen-antibody complexes. And that's something that's the result, again, of a protein made by a B cell, an antibody, interacting with a toxin that comes from a living or a non-living pathogen. Basophils make up about 1% of the total white count. They have a U-shaped nucleus with blue staining granules, and they move into tissues to mediate the inflammatory response. When they're in the tissues, they're called mast cells, and what they do is release histamine, and histamine is a very powerful inflammatory chemical. It tends to draw other white cells to the area. It promotes vasodilation. It promotes extravasation, which is the exit of white cells near the area of the injury. And this is all, again, designed to essentially rally your body's white cells to fight infection and to repair damage near the injury site. So this is, again, the role of histamine. It also releases a compound known as heparin, and this helps to inhibit clot formation. The idea behind this function is that in the area of injury, a cut or a puncture or what have you, we tend to have the formation of a solid clot in and around the damaged tissue. The basophils that enter this walled off area produce the heparin, which keeps the pathogens in a liquid state. It also enables the white cells to more effectively fight the infection. And at the end of the day, the heparin also serves to dissolve the clot once it's already served its purpose. So, a couple of different roles for heparin. A few other things about histamine. You've probably heard the term histamine if you've ever heard of a class of drugs called antihistamines. And what they do is they actually retard the activity of histamine, so they cut down on inflammation in the presence of chemicals like histamine. And what this does is it makes you feel better because it reduces the inflammation, but at the same time it provides a window of opportunity for the pathogen to prolong its infection cycle. So really, most of the time when you're taking anti-inflammatories, you're making a trade-off. Do I want to feel a little bit better, but have the pathogen linger longer inside my body, or do I simply want to have my body rally to the cause, use the inflammatory secretions to bring the white cells to the area, fight the disease, knock out the pathogen, and feel relatively ill, but for a short period of time, and then recover? That's the trade-off you're making when you take antihistamines. Now, sometimes inflammation isn't indicative necessarily of an encounter with a pathogen. Sometimes it's the result of an over-response on the part of cells that produce histamine, such as in allergic reactions. And in that case, you might see how the use of antihistamines would be warranted. Lymphocytes come in two flavors, B lymphocytes and T lymphocytes, and among the T lymphocytes, we have several subclasses. We have the cytotoxic T lymphocytes, which engage in a physical and chemical attack at the site of infected tissue or wherever the pathogen is located. We have regulatory T lymphocytes that turn off the immune response once the pathogen has been conquered. So cytotoxic Ts, regulatory Ts, and then with the B cells, we have really one flavor of B cell, and their job is to produce antibodies. Okay, so very important that we understand the different classes here. Another class of T cell we should touch on is the helper T cell, which really sits at a regulatory position in between the B and the T cell population. The helper T cell's job is to chemically stimulate the production of specific B and cytotoxic T cells, as well as regulatory Ts, that are designed to deal with a particular infection as a result of an encounter with a particular pathogen. So the helper T's job is to select and amplify those B and T lymphocytes that are best chemically equipped to destroy a particular bacteria or parasite or mold or what have you, while the other B and T lymphocytes that have different specificities, meaning that they're really designed to fight different strains of pathogen, they remain at their normal numbers. Lymphocytes make up about a quarter of total white cells. They're the smallest white cells. They have a spherical nucleus, no visibly staining granules. The two major types are the T lymphocytes that attack and destroy pathogens, and the B lymphocytes that generate the antibodies that go out and attack either a living pathogen or inactivate a toxin, whether it be a toxin that's actually attached to the pathogen or one that the pathogen secretes. Another way that the antibodies fight disease is through a process called immunoprecipitation or agglutination. And essentially what works here is that the antibody, and we'll draw an example of what an antibody looks like here. Let's use blue for fun. The antibody, most antibodies have a minimum of 2 binding sites on their surface. Antibodies are basically Y-shaped molecules that have a short chain and a long chain. The short chain is held to the long chain by disulfide bonds, and then the area here at the ends of the Y-shaped molecule, those are the antigen binding sites. And so the way that the antibody does this trick is, if we imagine, let's pick a different color for the pathogen. Let's make the pathogens red. If the pathogen is invading, okay, so these are pathogens all peppered throughout the body, and we, as a result of an encounter with a molecule on the pathogen's surface, select a particular class of B lymphocyte that's going to make an antibody designed to bind to this particular pathogen, basically to a molecule on the pathogen's surface. What will happen is that when these antibodies come into contact with these pathogens, they will bind them together, okay, in a big non-functional mat. Okay, and this is what we mean by immunoprecipitation or agglutination. In agglutination, we're binding together living cells. In immunoprecipitation, we're simply binding together non-living components. Okay, but the principle is the same. By having these antibody molecules come in here and attach to all this stuff, the molecular weight of the complex gets so big that it can't stay in the plasma, and the result is that it comes out of solution and is easy pickings for the next cell we're going to look at, which is the macrophage. Okay, so that's an example of, we'll call it immunoprecipitation. Immunoprecipitation or agglutination. Now, monocytes are very important because they work in concert with lymphocytes to help us fight disease. They make up, yeah, call it about 6 to 7% of the total white count in typical plasma volume. They have U-shaped or kidney-shaped nuclei. They do not have visibly staining granules, and their function is phagocytosis of bacteria and cellular debris. Basically, they come around, they eat stuff up. Once in tissues, we call them macrophages. Um, and they're important because not only does phagocytosis destroy the pathogen that's attacked, in addition, the the pathogen that gets endocytosed and destroyed when it fuses with a lysosome, bits of that pathogen get presented on the surface of the macrophage along with proteins called major histocompatibility proteins, and that serves as a warning to lymphocytes that we have an infection. And it's only going to be those lymphocytes that interact very specifically with those presented molecules on the macrophage that are going to be chosen to divide and generate an army that can go out and conquer and destroy this particular invader. The rest of the lymphocytes with different specificities will be left alone. Now, platelets, also known as thrombocytes, are cytoplasmic fragments of stem cells called megakaryocytes that are found in the red marrow. Um, and they're important because they regulate blood clotting. You've got 150 to 400,000 of these per cubic millimeter of blood. They plug breaks in blood vessels and they initiate the clotting process. So very important that you have an adequate number of platelets. They also lack a nucleus. They don't have cellular organelles. They don't have genetic material. In that way, they're similar to erythrocytes. All the other formed elements we've been talking about, all the leukocytes, they contain nuclei and organelles, and that's important because that helps them carry out their role in fighting disease. But for the platelets and for the erythrocytes, job number one is to transport a particular type of chemical around the body. In the case of the erythrocyte, it's there to transport oxygen and carbon dioxide using the pigment hemoglobin. In the case of the platelet, it's to regulate the clotting process by carrying around proteins that initiate and regulate the blood clotting process. If you didn't have any platelets, you'd have a difficult time controlling bleeding when you had an injury. You would tend to continue to bleed. And there's actually a disease in which that happens, which is called hemophilia. Now, if we were to take whole blood and spin it in a capillary tube, we would find really three layers. If we were to draw this, let's draw this. We'll draw the whole blood before and after spinning. So let's do here, let's do a black hematocrit tube. So we will draw that. So there's our hematocrit tube here. Okay. And then we usually draw blood up inside it. One end is generally sealed. And so this will be our blood in red. Okay, so the whole blood would look like this, all inside the hematocrit tube. And then we put this thing in a centrifuge. So we put this thing in a centrifuge. We spin it. And then when the tube comes out, you have several layers inside the capillary tube. So this is our capillary tube after the spin. There's our clay plug. So what you'll have is a layer of packed red cells. That represents the hematocrit. And it's not going to take up the entire volume of blood. Okay, it's going to be between 45 and 55%. And then on top of it, you're going to have a immediately on top of it, you'll have what we call a buffy coat, which is a layer of whitish debris, which is mostly platelets and leukocytes. So let's do that in gray. Okay, so there's our Buffy coat here. And then on top of that, you'll have the straw-colored plasma, and we'll do that in yellow. Okay. So there's our plasma on the top of the tube. And the reason that we generate these different layers is because the cells are denser than the liquid plasma. So when they're spun in the centrifuge, they're going to settle to the bottom of the tube, while the plasma is going to be less dense and stay on the top. It's 90% water, the plasma is, and it contains dissolved solutes and formed elements. It contains a lot of different stuff: proteins, ions, sugars, hormones, a fair degree of bicarbonate is found in the plasma. These solutes are kept in a state of dynamic balance inside the plasma. Plasma proteins are the most abundant solutes. They're generated by the liver. And there's 3 types of plasma proteins: albumins, globulins, and fibrinogens. The albumins are important because they help the blood plasma retain its fluid volume through osmotic activity. They're very large proteins. The globulins are important because they regulate our defense against disease. And fibrinogen is a protein that's involved in the clotting cascade. So this is how we generate a seal in a blood vessel or near a wound so that we don't continue to bleed after we've been injured. So albumins are 60% of our plasma protein. They also have a buffer activity. It keeps the pH of the blood between 7.35 and 7.45. They maintain the osmotic pressure of blood. A decrease in osmotic pressure in blood will cause water to move into tissues, and this causes edema. We sometimes see this in individuals that have a very protein-poor diet, or individuals that have liver failure, because in both of those situations, we're reducing the albumin content of the blood plasma, which means that the blood can't hold on to as much of its water as it would like to. As a result, we see a decrease in blood volume and an increase in interstitial fluid volume. An increase in osmotic pressure in the blood is going to cause water to move into the blood and cause the blood volume to increase. Okay, the albumins are so large that they have a difficult time moving across the walls of capillaries or blood vessels. They tend to stay in the plasma. But other components that are osmotically active, as we'll learn in a bit, such as ions, can move through cells with the aid of plasma proteins pumps. And this is actually one of the ways that the body directs where the fluid goes, is by pumping ions into different fluid compartments in order to maintain our water balance between the plasma, the lymph, the interstitial fluid, and so on, all the fluid compartments in the body. Okay, so an increase in osmotic pressure in the blood is going to pull water out of the surrounding tissue, and the result is that the tissues themselves will dehydrate a bit. Okay, globulins make up a little over 1/3 of the plasma proteins. The alpha and beta globulins carry lipids and fat-soluble vitamins in the blood, while the gamma globulins are antibodies that are involved in immunity. Okay, so they help us fight disease. Fibrinogen makes up about 4% of the plasma proteins. It has an important role in blood clotting, and it's converted into fibrin when blood clots form. Okay, so very important to understand the role of fibrin. If any of the blood clotting proteins are absent or defective, then we will continue to bleed long after we experience a penetrating injury. And an example of the condition like that is hemophilia. Okay, now what else do we find in the blood? Well, we also find non-protein nitrogen-containing substances such as amino acid and urea and uric acid. These are waste products of protein and nucleic acid breakdown. Amino acids, remember, are the building blocks of proteins. The urea and the uric acid tend to be excreted in the urine, and the reason for this is they're bases. Okay, what bases do to the pH of tissue fluid is it causes the pH to go up. So if we were to allow these waste products to continue to accumulate in the tissue fluid or in the blood plasma, after a while, what would happen is the pH would be changed so dramatically that we would perturb membrane potential and we would denature proteins and we would probably slip into coma and death. Okay, so the body takes great care to keep our nitrogen balance within homeostatic parameters. We generally consume as much nitrogen as we excrete, with just a very few exceptions. Okay, so very important to understand that. Electrolytes are ions that are absorbed from the intestine or released from cells. Most common ions are sodium, potassium, calcium, chloride, bicarbonate, and phosphate. Now notice that some of these ions have a positive charge, so those are the cations, and some of these ions have a negative charge, so they're called the anions. They help maintain osmotic pressure in the blood and in tissue fluid, and they also help to maintain the plasma pH. Okay, our next topic is hemostasis. Now, what is hemostasis? Hemostasis is basically the ability of the body to stop bleeding in the face of an injury after a period of time, right? Hemostasis involves three processes: blood vessel spasm, which is mediated by smooth muscle constricting right after the injury; platelet plug formation, which is basically where the thrombocytes block the area of the injury; and blood clot formation, which is the formation of a fibrin clot which entangles and enmeshes other formed elements in order again to form a seal between the inside of your body and the opening that's produced by the wound. Okay, so we have to keep you from bleeding out. So vascular spasm results from the constriction of a damaged vessel. There are factors that are released by damaged tissue that promote smooth muscle contraction, and so this is a temporary cessation of bleeding. Serotonin from the platelets contributes to this. The plug is essentially a congregation of platelets that are attracted to the damaged site and attached to the exposed connective tissue. Collagen fibers, for example, are one of the things that snag thrombocytes and cause them to adhere to each other. This also results in platelets releasing their clotting factors that they contain, and that begins the whole process of generating a fibrin clot. So a couple of things go on, right? We spasm, then we make that platelet plug, and then we promote the formation of a blood clot. And here is what the process looks like from injury to the beginning of the healing process. Here you've got the damaged vessel. The collagen fibers get exposed. Blood escapes through the break during the initial portion of the injury, and then the platelets stick to each other and to the ends of the broken vessel and the collagen, and then eventually what happens is that they plug the hole and then begin to release their clotting factors, which are going to promote now eventually the production of a fibrin clot, which will serve the purpose of closing the hole and allowing now cells that are going to come in and try and bring the damaged ends of the tissue together and then ultimately promote cell growth so that the injury can heal. Now, coagulation is the formation of a blood clot. It's a complex process that involves a series of proteins that operate in what we call a cascade. Blood has procoagulants that promote clotting and anticoagulants that inhibit clotting, and they normally are operating unless we have an injury. When we have an injury, when we have damaged tissue, the procoagulant activity initiates the clotting process, It usually completes within 3 to 6 minutes, and the clots are normally restricted to the area of the damage where procoagulants predominate. After clot formation, the fibrin, which is the protein component, the most significant protein component of the clot, contracts and pulls the damaged tissue closer together. The fibroblasts migrate into the clot and they begin to destroy dead tissue. And in addition, there are chemicals that promote cell division that are going to ultimately result in the closing of the wound. Tissue plasminogen activator is part of the clotting cascade. It converts plasminogen into plasmin, and the plasmin ultimately breaks down the fibrin and dissolves the blood clot after it's served its purpose. Okay, so we have an initial set of reactions that results in the conversion of fibrinogen to fibrin, and that's the last stage in forming the clot. And then in the context of the clot, what we do is we battle the pathogen, we remove the dead and dying tissue, and then we promote the contraction of the wound together. At the same time, we also promote cell division in order to close up the area of the injury. Okay, and once that's happened, we don't need the fibrin clot anymore, and that's when the plasmin comes in to do its trick. Okay, next thing we want to talk about are blood types. Now, all blood is the same functionally. Okay, all blood is designed to transport materials around the body, among them oxygen, carbon dioxide, hormones, nutrients, and waste products. All right, and all human blood has the formed elements that we've spoken of, right? That all contains thrombocytes, erythrocytes, and leukocytes. When we talk about blood types, what we're really talking about are chemical ID tags that are on the surface of red blood cells that are there in order to indicate to your immune system that those tissues are your own. Okay, so it's essentially your body's one of your body's ID tags. So what are these molecules, these ID tags? Well, for the ABO groups, we're going to find out that they're clusters of sugars that are on the surface of the cell membranes of the erythrocytes. And for the RH group, we're going to find out that it is a protein that's embedded in the plasma membrane. And they're both products of different genes. Okay, so they're not related to each other in the sense that one depends on the other. They can be expressed independently of each other, and this is what gives us the variety of blood types that we see. An antigen is a special molecule on the cell surface. It could be a protein, it could be a carbohydrate, it could be a glycoprotein, which is a combination of protein and sugar. It could be a lipoprotein, and there's all kinds of things that can serve as antigens. The common blood types we're going to talk about are the ABO groups and the RH factor. Antibodies in the blood will not recognize antigens that are, antibodies in the blood will recognize antigens that aren't found normally on your tissues. And this is really how the immune system discriminates what to leave alone and what to attack as if it were harmful to the body. It discriminates based on self versus non-self. The agglutination occurs when the antibodies bind to the foreign antigen and cause the foreign red cells to clump together. Transfusion is done when an individual is going to experience blood loss. So what we often do is give people whole blood, or in many cases we give them packed cells resuspended in saline solution in order to compensate for blood loss, say during a surgical procedure or in the event that they have an injury that causes them to hemorrhage. The blood is usually of the same type. It's very important that we ensure that the recipient's blood and the donor's blood are compatible, and then we call that typing and cross-matching. If they're not, what will happen is that the recipient will experience hemolysis, as the antibodies present in their plasma attack and destroy the donor blood, causing it to lyse, releasing hemoglobin into the plasma where it does not belong, and resulting in the blockage of capillary beds and the necrosis of tissue downstream from the block. Okay, so that's gonna kill somebody. So it's very important that we type and cross-match properly. So ABO blood groups depend on the presence of antigens. There's an A antigen, a B antigen, and there's also an O antigen. But only the A and B antigens have corresponding antibodies. The O antigen is not antigenic enough to promote the formation of an antibody against it in normal human whole blood or serum. Okay, so there's 4 blood types that are possible: A, B, AB, and O, and they're all based again on collections of sugars on the plasma membrane of the erythrocytes. Remember that any person's blood, whole blood, is not going to possess antibodies against their own antigens. Otherwise, they would hemolyze their own red blood cells and they would die. Okay, so what I want you to do is watch the video on type 2 hypersensitivity and how blood typing works. And I'll join you in just a second. So typing blood involves the application of anti-A and anti-B antibodies to drops of whole blood, and what we look for is agglutination, which is the clumping of cells in the presence of antibodies that react to the surface antigen. If agglutination takes place, we know that that antigen is present on the surface of the red blood cell. When we transfuse blood, antigens in the donor and the recipient blood have to be compatible, and this ensures that agglutination won't occur with the donated red cells. So if we look here, we have an example of, in the top panel, a type A blood donor, and anti-B antibody in the type A blood will react to anything that contains the B antigen, okay? But the antibody will not react to the A antigen of the type A blood, and of course the O antigen does not have a corresponding antibody. Now, below, you can see this type A blood donor receiving now blood that contains the type B antigen. This could be type B blood or it could be type AB blood. Both contain the B antigen. If we were to do that, we would kill this type A individual because the antibodies already present in the serum would agglutinate and hemolyze the red blood cells. The result would be rupture of the erythrocytes, and the result would be massive crisis. Okay, the patient would likely die in this event. Type AB blood is considered to be universal recipient because in type AB blood there aren't going to be antibodies against either the A or the B antigen. The result is that that individual can receive any blood type. Okay, it possesses no antibodies that will react with donor blood. As a result, we don't have to worry about the donated red cells. Now, this is apart from the Rh factor, which we'll talk about in a minute. In type O blood, what we have is universal donor. Now, you might ask, well, how can type O blood be universal donor? Well, remember that the O antigen does not have a corresponding antibody, so we can give type O erythrocytes to any recipient, and we don't have to worry about agglutination and hemolysis. But you might say, well, wait a minute, what about the antibodies in the type O blood? And that's a good question. What we generally do when we collect blood is we don't store it as whole blood, and we don't give it to the patient as whole blood when they need a transfusion. What we do instead is we remove the red cells, and we separate the red cells from the plasma and resuspend the red cells in simply a saline solution that's isotonic so that the red blood cells won't rupture. As a result, the resident plasma antibodies aren't there, okay? And as a result, they're not going to react with the recipient's blood, okay? So we give them packed cells and saline. We don't worry about that. So the only thing we concern ourselves with in a transfusion is the recipient's antibodies and the donor blood's antigens. That's why. Okay, the Rh factor is again another surface antigen, sometimes called the D antigen. Rh comes from the fact that it was discovered first in the rhesus monkey. If the antigen is present, you're said to be Rh positive. If the antigen is absent, you're said to be Rh negative. If anti-Rh antibody is added to blood and agglutination occurs, the Rh antigen is present. Rh negative individuals do not normally have significant amounts of the anti-Rh antibody in their plasma. They only have significant amounts in the plasma if they've been exposed to the Rh antigen as a result, say, of a mismatched transfusion where they accidentally got Rh positive blood, and what happens is that exposure will result then in the generation of anti-Rh antibodies, or in the case of an Rh negative mother who's carrying an Rh positive child. At the point where the child is born, the placenta is torn from the uterus, the baby's blood and the mother's blood mix temporarily, and if the baby's Rh positive, the mother's Rh negative, that will constitute an exposure, and the result then will be that the mother will begin to produce significant amounts of anti-Rh antibody. So it's formed only after exposure to this antigen in Rh negative individuals. The first exposure builds the antibodies without agglutination, but subsequent exposures will result in agglutination and could potentially kill the individual that receives, the Rh negative individual that receives the Rh positive blood. Erythroblastosis fetalis is a disorder in which newborn infants result in the destruction of the fetal red cells by maternal antibodies. Now, the way this happens is that an Rh negative mother has an Rh positive child, a first Rh positive child, and at birth she's exposed to the Rh positive blood, and then in between the first and second pregnancy she begins to develop anti-Rh antibody. The anti-Rh antibody has to be small enough to cross the placenta, so while the child's developing in the mother's womb, there's the potential for the antibody to cross into the baby's blood and agglutinate and hemolyze it. Okay, and that can, in some cases, kill the child. So, an Rh negative woman pregnant with her first Rh positive fetus, that pregnancy will go fine. Okay, there won't be any problem. But the mother will begin to develop Rh antibodies in the interim between the first and second pregnancy with an Rh positive child, and the result then in the second pregnancy is that her antibodies can pass through the placenta and destroy the fetus's red blood cells, okay, and potentially kill the baby. Now, what do we do about this? Well, we have a drug called Rogam. Okay, now don't confuse Rogam with Rogaine. Here, let me spell Rogam for you. Do it in black. Okay, talking here about Rogam, R-H-O-G-A-M, M as in monkey. Okay, now what is Rogam? It's essentially a compound which masks the Rh antigen in the mother's bloodstream. The result is that individuals, Rh negative women with Rh positive babies, if they're on Rogam, at the point at which the baby's born, if the Rogam is present in sufficiently high levels, that'll block the Rh antibody from detection by the mother's immune system, and then she can have as many Rh positive children as she wants. Okay, so this is one of the things that we do in women who are trying to become pregnant, is we do a blood test to determine Rh compatibility between the husband and the wife. If the man is Rh positive and the woman is Rh negative, there's generally a 50% chance that on any given pregnancy they can have an Rh positive child. And if that's a possibility, then what we do is we start the mother on Rogam before, during, and after the pregnancy. The result then is that the fetus and the neonate are protected. Okay, it's not an issue if both the male and the female are Rh negative, because there's no possibility of generating an Rh positive child in a marriage like that. It's also not a problem if the male is Rh negative and the mother is Rh positive, because the mother, again, is never going to develop antibodies against her own blood. Okay, so it's only an issue when the mother is Rh negative and the father is Rh positive, as shown in the diagram. Fetal red cell destruction in this event will reduce oxygen carrying capacity of the blood. This releases immature red cells into the blood. They can't carry oxygen like mature red cells. In addition, the hemoglobin that's released blocks capillary beds, and this interferes with kidney function. It cuts off blood supply to downstream tissues. Bilirubin buildup can cause jaundice, and low oxygen and high bilirubin can result in brain damage and death. Okay. Afflicted infants can be treated by replacing total blood with Rh negative blood. This adds functional red cells back to the infant's blood supply. It clears out the bilirubin, but that's really an emergency situation. We try and avoid that altogether, again, by having the ROGAM present before, during, and after the pregnancy. Okay, let's talk a little bit about some blood disorders. They can be grouped as erythrocyte, leukocyte, or clotting disorders. So this is an example of an erythrocyte disorder. This is anemia. It's a reduced oxygen carrying capacity of the blood due essentially to a lack of red blood cells and thus a lack of hemoglobin. It can be caused by a decrease in red count or insufficient hemoglobin in the red cells. And so there's different types of anemia. Nutritional anemia is the result of a lack of iron or B12 in the diet. These are important building blocks for red blood cells. Okay? The iron, of course, is a component of hemoglobin. The B12 is an important component of DNA. Okay? It supports certain metabolic pathways that build nucleic acids, and the hemocytoblasts that produce the erythrocytes rely on this in order to continue to generate erythrocytes. Hemorrhagic anemia is blood loss through excessive bleeding, while pernicious anemia is generally due to a lack of a protein called intrinsic factor that escorts the B vitamins across the gastric mucosa and into the bloodstream. So here, it may be present in the diet, but if you can't get it in the blood, it's not going to do any good. Hemolytic anemia is premature rupture of red cells, while aplastic anemia is the destruction of red marrow or its inability to produce enough red cells. So there's all types. Sickle cell anemia is a genetic disorder that results in an abnormal hemoglobin that, under low oxygen concentration, forms an unusual shape. It tends to polymerize inside the red cells that carry it, and the result is that the cells take on this sort of crescent moon shape that ends up sticking in capillaries and cutting off oxygen to downstream tissues. If you have two copies of the sickle cell gene, the result is going to be sickle cell anemia. If you have one copy of the sickle cell gene, you're going to have what's called sickle cell trait. And so in conditions of extremely low oxygen concentration or high intensity activity, you might experience some hypoxia. Okay? So this is more common among certain ethnic groups, racial groups. In its heterozygous form, if you have one copy of the sickle cell gene, it actually promotes resistance to a blood disease known as malaria, because the parasite that lives in the blood can't complete its life cycle in individuals that carry the sickle cell gene. The problem is that we took this gene out of its normal location when we invented planes, trains, and automobiles, and it doesn't serve us well at high altitudes or in areas of the world where malaria is not endemic. Erythroblastosis fetalis, we discussed earlier. It's the destruction of fetal red cells that are Rh positive by the mother's anti-Rh antibodies. It causes anemia, jaundice, and the presence of erythroblasts in fetal blood. Polycythemia is an excessive number of red cells. Now, sometimes this is an adaptive response to low oxygen conditions, but sometimes it's the result of disorders of the bone marrow. It increases blood volume and viscosity, and this can impair circulation, and it can be due to cancers that affect the hematopoietic tissue. Okay, leukocyte disorders. These are your white cells. Mononucleosis is a contagious disease caused by the Epstein-Barr virus. It's primarily found in young adults. Kissing is a common mode of transmission, and it affects the B lymphocytes. The symptoms include fever and sore throat, and it lasts 4 weeks to months to years. Viruses are particularly difficult to deal with because they're essentially non-living pathogens that produce their effects by invading living cells and using our own reproductive machinery against us. And so generally, in order to kill a virus, you have to kill the virus's host, and that's very tough to do. So there's very few effective antivirals out there because the virus doesn't provide a lot of good drug targets like bacteria, parasites, and mold do, because they have a biochemistry that's distinct from our own, and as a result we can design drugs that will affect those organisms, which are free living generally, and not disturb our own body function, whereas in a virus, the virus is already inside us. It's already inside our cells. Very difficult to get rid of. Leukemia is cancer of the white cells, particularly those cells that form white cells in the bone marrow. Excessive white cell production inhibits red cell and platelet production. You can have acute forms in children and adults and chronic forms in adults. The treatments include chemotherapy and radiation, just like we have for all different types of cancer, and bone marrow transplant. You might think, well, why are these individuals experiencing a problem? They got all these white cells. Aren't they going to have all this wonderful disease-fighting capability? Well, the problem here is that the white cells that are produced in leukemia are not effective at battling pathogens. Okay, they're made in improper fashion, and so essentially they impair the ability of the normal white cells to do their job. Let's say clotting disorders. Hemophilia is an inherited disease. It's more common in males because most of the hemophilia genes are on the X chromosome. Remember that men only have one X chromosome while women have two. The Y chromosome has no traits on it other than the trait of being male, and as a result, it's more common in males than it is in females. Females would have to have two bad copies of a hemophilia gene in order to show the disease, while males only need one. Okay. What we see in hemophilia is spontaneous bleeding and reduced ability to clot blood. This is due to the lack of a clotting factor or a defective clotting factor. The treatment involves transfusion of the missing clotting factor in order for blood clotting to take place. Thrombocytopenia is a lack of platelets, and this causes spontaneous bleeding to occur. We're not capable of generating the fibrin clot quickly enough, and the result then is that we tend to bleed more extensively than we would if we had those clotting factors, or if we had those thrombocytes present. A thrombosis is the formation of a clot in an unbroken blood vessel that cuts off blood to downstream tissues, specifically near areas that are roughened or damaged. If this happens in an artery, the tissues that are deprived of oxygen will die in relatively short order. Thrombophlebitis is an example of this happening in the lower extremities. An embolus is simply a traveling clot that's moving through the circulatory system. Eventually it's going to lodge in a blood vessel that's too narrow to allow it to pass, and the result will be the same as if we had a thrombus. Essentially, you'll cut off blood supply to downstream tissues. The result is going to be death of those tissues. One of the things we have to understand about clotting disorders as well is that it's not simply damaged tissue that can promote clot formation. It's also hemostasis, basically lack of blood flow. What happens in individuals that aren't active is that blood can pool, and one of the things that happens when blood flow is impeded is that clotting factors transiently concentrate in the plasma and promote clot formation, and the result then is that you can have an embolus that'll travel and ultimately lodge in a capillary bed and cut off circulation to downstream tissue. So this is one of the reasons why we always tell people try and remain active, okay? Because the more active you are, the more blood flow you have, the less likely you are to end up with these emboli, okay? All right, stay tuned for some videos on blood, okay? Talk a little bit about the components of blood and some blood disorders, and I'll see you guys in the next podcast. Thanks for listening.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin, to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.